So, good morning, students. Today we are going to discuss a chapter, a fourth chapter in biobotany as well as the botany, the principles and application of biotechnology. It's one of the important topic for your examination point of view. And there we are going to discuss one small important topic. It is very important for your NEET exam as well as it's very important for your board exam. Okay. So I'll give the topic, name of the topic, insertion, inactivation, a blue and white colony selection method. So it's a 4.6.1, I think so. Okay, am I correct? I think so, right? So this topic is a very important for your examination point of view, particularly for the NEET exam. It's very important. So what is insertion, inactivation, blue and white colony selection method? So it is one of the most powerful method in which you are going to screen a recombinant plasmids. Recombinant plasmids. You know what is a plasmid? Okay, the plasmid is otherwise called vector. You know what is a plasmid? Do you know where the plasmid is present? Of course, the plasmids are present in the bacterial cell. Okay, even the E. coli, which is living in the human body, they contain plasmids. Plasmid is nothing but it's an extragenetic material of the bacterial cell, which is found in the cytoplasm of the bacterial cell. It's extragenetic. Already it's having its own DNA, but along its own DNA, they possess some extra genetic material. This extra genetic material is called a plasmid. So define a plasmid. Suppose they can ask in the board exam. So plasmid is nothing but it's a small circular double-strand self-replicating DNA molecule. Okay, that's a plasmid. So plasmid is otherwise known as vector. Right now, in this method, I am going to insert a LAC-Z gene. It is a reporter gene. LAC-Z is a reporter gene. So this reporter gene I am going to insert in the vector. Okay. Now this LAC-Z reporter gene is responsible for encoding an enzyme called beta galactosidase. Look at this. So beta galactosidase. Beta galactosidase. So this enzyme encoded by LAC-Z. Okay. So what is the role of this enzyme? So this enzyme responsible for to break down this synthetic medium, artificial or the synthetic substrate. Xgol is a synthetic substrate. The full chemical name of Xgol is 5-bromo-4-chloro-indolyl beta, D galactopyranoside. So it is a synthetic substrate. So this enzyme, beta galactosidase enzyme, break down into insoluble a blue colored byproduct, a blue colored product or a blue colored byproduct. That's the role of this beta galactosidase. Okay, that's why we can see the Xgol. Xgol is the white portion. The white portion is Xgol. So this Xgol is break down into insoluble blue colored byproduct. Yeah, see, look at this blue colored. Here, lot of blue colored are spreads. Okay, you can see there are lot of blue colors. Right. Now we'll go to the next one. That is, now I am going to insert a foreign DNA. I am going to insert a foreign DNA into the LAC-Z gene, a reporter gene. Okay, here the reporter gene, LAC-Z gene. I am going to insert, I am going to place a foreign DNA. Okay, this is in Tamil I have written, it's a foreign DNA into the LAC-Z gene, which results the LAC-Z genes get inactivated. Okay, by inserting of the foreign gene into the LAC-Z gene, the LAC-Z genes gets inactivated. This is called a insertion inactivation. Okay, so they get inactivated. So how they can get inactivated? How can I know the lac acid genes get inactivated? That's why you take that plasmid or vector, you place in the X gall. Okay, so they are unable to produce blue color colony or blue color byproduct, insoluble blue color. Why? What is the reason? Okay, because the lac acid gene, a reporter genes, gets inactivated so that they do not produce beta galactosidase enzyme. So they produce the white colored. Here, right. Now I have the two cells, two host cells. I don't know which cells have the recombinant DNA and which cells do not have the recombinant DNA. Okay, so I am taking these two host cells into the X gall. Okay, X gall. Suppose the cells which possess recombinant DNA, they produce white colony. Why? Because the I am inserted the foreign DNA into the lac acid. Okay, that's why they do not produce blue colony, they produce only the white colony. Okay, the another cell in the X gall, they produce blue color colony because the cells which produce a blue color colonies, they are non-recombinant. Okay, non-recombinant because they do not have the recombinant DNA in their cells. They do not have the recombinant DNA in their cells, so they produce a blue color colony. Okay, so by means of these colors, blue and white, okay, the cells, the host cell which produce a white colony are called recombinant DNA. They possess a recombinant in their cells. Okay, the cells which do not possess DNA, recombinant DNA, they produce blue color colonies. So these blue color colonies do not have the recombinant DNA. So by seeing these colonies white and blue in the X gall, so I can able to screen. Okay, which is recombinant DNA, which cell is possess recombinant DNA, which cells do not have the recombinant DNA. Okay, right. By means of color, you can able to identify or you can able to screen the recombinant plasmids. Okay, right. Thank you. So next class we will see one small topic.